Welcome, everyone, to this conversation with Stéphane Bancel. We're going to talk about AI and biopharma. We're going to talk about Moderna. We're going to talk about the future of medicine, treatments, diagnostics. Let me start with a question for the audience. My first question to the audience is, how big in trillions of dollars is the biopharma sector globally? How big? I'll start the bidding at $1 trillion. Any other bids? Any other bids? 3 trillion, I heard? 4, 5, it's going up. So it's between 8 and 10 trillion dollars, growing very, very fast. And the question is, what do we get for those 8 to 10 trillion dollars? Today, Stéphane and I will talk about how biopharma is working today, some of the big breakthroughs that Moderna has had specifically, and we'll talk about where we can take this and revolutionize the biopharma industry. So let's start. By the way, please keep up because there'll be more questions for the audience in a little bit. Stéphane, let's actually start with you as a person. Your father was an engineer, your mother a physician, you grew up in France. How did the professions of your parents, engineer, physician, shape your choices, your life choices, your career?
Well, good afternoon and thank you. I'm super happy to be back in the kingdom. Well, given I'm a chemical engineer and actually a biochemical engineer, I think you get the answer to your question. Big impact from both of them. But also my education, I was raised by the Jesuits, so really a sense of serving was important, which is why I spent all my life in healthcare. But also mentors on the way. I've been lucky, like many of us, to get a lot of great mentors. Alain Mérieux, chairman of Biopharma, and I was CEO of Biopharma, taught me a lot. And our chairman, Noubar Afeyan at Flagship.
Noubar Afeyan,
yes,
head of Flagship.
Has also helped
here at
me think bigger as we built Moderna.
Wonderful. And in the early part of your career, you actually joined Lilly in different positions. Lilly obviously was a great company then, and even a greater company now, with Mounjaro and many other breakthroughs they've had. I think they're reaching $1 trillion in market capitalization recently. Tell us about your experience there. What were your takeaways from your experience at Lilly, and what are you using now, lessons that you learned then?
Sure, so a lot of lessons in terms of scale, managing scale, global businesses, integrating and making sure that you have your R&D, talking to your manufacturing, talking to your commercial. But I think the piece that I was most surprised and sometimes shocked by in big pharma is the failure rate of drugs. As you know, highly documented, 90% of the drugs that start a phase 1 never get approved, which if you think about it from an ROI standpoint, is just terrible.
Let's just emphasize that, please, Stéphane. 90% of drugs, of candidate drugs that go into clinical trial, never come out again. 90% failure. So if we were talking about, for example, the StéphanoJack construction company that builds hotels, and we told there's a 90% chance of it falling down the first 30 days, that would not be a great construction company. But that is biopharma today.
That has been for 150 years. And one of the things that I love about the concept of Moderna 10 years ago when we started the company is because we use mRNA, which is an information molecule. If you think about the pharma industry for 150 years, it has been an analog business where every molecule is different. If you look at a Prozac or a Lipitor or pick your favorite pharma drug, it's a unique chemical compound where you have to relearn everything in the lab, how to make it manufacturing-wise, prove to the regulator it's safe and it's working, which is why the success rate. It's a trial and error process where everything has to be relearned every time. But if you think about mRNA, as we all learned during the pandemic, it's a molecule made of 4 letters. And every mRNA product we make is the same 4 chemical letters we put in your body. And so because of that, we have already showed that Moderna, our success rate from a phase 1 start to a phase 3 successful data is 6 times the industry, 6x. And I think over time, it has a chance to get even better.
And why do you think you're getting this massive success rate compared to the benchmark in the industry?
For 2 reasons. One is toxicity, because it's always the same 4 letters that constitute an mRNA molecule. We put the same chemical in your body. Those are chemicals anywhere that exist in every one of your cells. Those are not like foreign chemicals. Again, think about a Prozac or a Lipitor. Those are molecules that do not exist in your body. mRNA is in every one of your cells. The 4 chemicals that make the mRNA are in every one of your cells. So the toxicity side of the house is taken care of because it's always the same molecule for all of our products and the molecule that exists in nature. The second part is we code a protein that exists in nature. Think about the COVID vaccine. We did not invent spike. We copied and pasted spike when the virus was sequenced and put online by the Chinese government early in the pandemic.
You did that within weeks, right?
We did like in 48 hours.
There you go.
And so, and all in silico. We never had access to the physical virus to design the product. And the product that our team designed in silico on a weekend, just on the computer, is exactly the same molecule that you and I and many other people have gotten around the world. And that's the power of technology, is we use always the same brick to make the molecule, and the molecule always codes for something that exists in nature.
So let's talk about mRNA a little more, and let's go beyond vaccines. So just to remind us all, mRNA, you're sending in basically into the body a set of instructions that then make something else, a protein, that will then do something in the body. So it's a very innovative way of approaching treatment as opposed to just sending in the actual treatment. This is a set of instructions that we use the machinery of the cell to actually produce what we want. Obviously, Moderna became very, very famous during the COVID epidemic, the vaccines. You've saved millions of people's lives. But now the company has definitely still doing a lot of vaccine work, but also now moving to therapeutics as well. Let's talk about that, and particularly in oncology, in cancer. We all know people who are affected, impacted by cancer in every one of our families, friends, and circles. And so what cancers you focused on? I think one of the first we could start with was melanoma.
Mm-hmm. So we are focusing on an approach to teach your immune system to recognize your cancer that it missed. We all have cancer all the time. I maybe have cancer cells developing in my body today. But if I'm healthy, which is why the importance of prevention and sleep and nutrition and exercise, if I'm healthy, my immune system is going to see cancer when it's one cell, and it's going to eat it up. So it will never become a tumor. But if for whatever reason that immune system cannot see the first cancer cell, it becomes 2 cells, 4 cells, and then you get a picture.
And Stéphane, let's just say on that point, to emphasize your point about how our immune system actually is able to suppress a certain amount of cancer, we know that when you give immunosuppressive drugs to a transplant patient, within 2 years of giving those immunosuppressive drugs that push down our immune system, there's a 30% increase in cancer, showing your point that our immune system is suppressing quite a bit of cancer, but of course, sometimes it does run away.
Correct. And so what we figure out is a technology where we basically take a biopsy of your cancer, we next-gen sequence it, meaning we read every letter of a 3 gigabyte of letters of your DNA of a cancer cell. We do the same thing for a healthy cell. We upload both on AWS, and we compare every base to see where the mutations happen. Where did your cell go wrong? And what we do is we pick those 34 mutations that are the most relevant through an algorithm, and we build an mRNA molecule just for you or just for me.
Let's emphasize this. This is now the wonderful personalized medicine we've been talking about for 30 years in the healthcare industry. You're making that for that individual.
It's even more than personalized, it's individualized. Personalized for me is you and I get the same drug if we have the same biomarker, but it's an off-the-shelf drug. We don't give it to everybody, we give it only to a population that has a specific biomarker. In our case, it's individualized. Meaning if you and I are diagnosed today in a hospital in Riyadh with skin cancer by the same doctor, Moderna will make for you and for me a different chemical molecule. It will be different mutations from your cancer for you, for my cancer for me, that will inject is an intramuscular. And we have shown now with 3-year survival data, 66%, meaning 2 out of 3 people, 3 years after the treatment, have no distant metastases, which is the best leading indicator of survival. Because as we know, it's rarely your initial cancer that kills you, it's metastases, of course.
So let's really put this in context. Melanoma is a deadly disease before the advent of checkpoint inhibitor, which is immunotherapy. Again, an approach that does not use chemotherapy. It's not trying to kill the cell directly. Immunotherapy is a judo kind of drug that uses our own body's immune system, protects it through work of Merck, for example, with Keytruda, and Bristol Myers Squibb with their particular drug. And you are working in combination with these drugs. The difficulty of those drugs without your intervention is that only about 30% of melanoma patients are helped on a 1-year durable basis. So the question is what to do with the rest of the people, 70%, who are not helped by those initial drugs. And those drugs, by the way, again, miracle drugs, millions of lives saved, and actually also very, very positive for Merck and Bristol Myers. But you come along and say, okay, in addition to taking Keytruda or the BMS drug, take this literally individualized medicine. I think that's a good distinction between personalized and individualized. And we can save many, many more lives. So you're starting with melanoma, which obviously affects literally millions of people around the world. What other cancers do you start seeing now in your pipeline?
Sure. So we've already started the clinical study in phase 3 for the lung, also bladder, and we're going to be adding more and more tumor types. One of the other things that we're very excited about is the growth and the improvement in liquid biopsy, which, as you might know, Jack, is the ability in blood to find a few pieces of cancer cells very, very early in disease. Because as we know, if you can go after stage 1 or stage 2 cancer, your success rate is much, much higher than the stage 3 or stage 4 cancer.
So, Stéphane, let's talk about that with the audience. So, cell-free DNA, cfDNA, is one of the aspects we're leveraging in this super early detection, in doing a liquid biopsy. In other words, when we're saying liquid biopsy, we're not literally taking a biopsy like you would cut something out from the skin. We're taking a blood test, and then what do you find in that blood test? What are you hoping to find?
So what people are able to do, and this is not a Moderna technology, but a technology we are very excited about to complement our approach, is basically you're able to find through sequencing the blood sample, you're able to find cancer DNA that is different from your healthy DNA, and that's how they discovered actually the technology by accident. So that gives me a lot of hope, because if we could find people in stage 1 and stage 2 for some cancers that are today found mostly in stage 3 and stage 4, you will increase the survival rate tremendously.
Yeah. So that's also very promising. Treatment we talked about with immunotherapy, and now also early stage diagnostic. Let's finish up, Stéphane, with the topic of AI and biopharma, even beyond just Moderna. And when we think again about the dismal statistics, we talked about a 90% failure in clinical trial. I'll give one last question to the audience, which is, in an average year, how many new drugs are approved that are first in human? Not a Me Too drug, not another statin, but a first in human drug that hits a disease that had no drug before. I'll start the bidding at 1 new drug a year. Anyone? So on average, there's only 12 new drugs a year approved by the FDA in Europe and the Middle East and other major countries that are first in human, hitting a disease that we had nothing for before. Only 12 a year. Obviously, we're not doing well enough, given the thousands of indications and thousands of conditions out there to be treated. So when you think about AI for biopharma, both in the preclinical phase of preparing the target, validating the target, doing drug discovery, drug development, then in the clinical trial phase and beyond, what are you most excited about in applying AI to this area?
Sure. If I think about healthcare in general, I think there are going to be many waves of innovation thanks to AI. I think the first obvious one is helping doctors and patients better diagnose disease early. If you guys have not done, just upload your labs on ChatGPT and start having a dialogue with ChatGPT. It's quite interesting. If you look at diagnostic technology, I think imaging has been massively reported. There were now AI systems start to find disease, including cancer, much easier than human doctors looking with their eyes at radios and stuff like that. So I think it's going to be a very interesting space. Also in terms of democratization, taking costs down, and be able to take this to people who cannot afford healthcare. I think it's going to be really interesting. In terms of making drugs, I think the first place is accelerating clinical development, because it's basically hundreds of business processes that companies are running with data. When you put those two things together, you say, okay, that's going to be quite interesting. So we're starting to do a lot of things within the industry and at Moderna. For example, picking a dose of a drug. This used to be a process taking weeks and weeks with committees and committees. Now you can load all the data of your studies, clinical studies, into a GPTs which we built, and you can basically have a system tell you the dose in literally 2 to 3 minutes. So it's quite interesting, time-saving. You can do the same in terms of your interaction with regulators. When you file to the FDA, let's say, you file a gigantic amount of data, and then you start to get questions. But not 1 or 2 questions, you say hundreds of questions. And so today or yesterday, humans would go into the files to pick the exact answer.
Now we can do something much more revolutionary.
Now you just do a GPT, and you have a human still reading it to make sure, because we have to sign it, that what goes to the regulator.
So we still keep humans in the loop.
at this stage.
We use AI
Correct.
to accelerate.
To provide the work. So there's a lot of examples like this in development. In discovery, we're starting to invent new molecules using AI. If you think about the ability to go after big data, big scientific data in a lot of different animal systems, from mouse, rat, monkeys, and more, human data, and try to combine everything, no human has the ability in their own head to look at the hundreds, in our case, thousands of animal studies we have done since the beginning of the company.
Yeah.
None of my scientists can do that.
Stéphane, pleasure to be with you. Stéphane Bancel, please give it up. CEO Moderna. Thank you very much.